Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuh. In our previous lectures, we have discussed different NMR techniques, that is proton NMR, carbon-13 NMR, cozy, nosy, etc. And we have come to know how these techniques have been helpful in elucidating structure of organic molecules. Today we are going to discuss another technique, that is HMBC. HMBC stands for Heteronuclear Multiple Bond Correlation Spectroscopy. As the name indicates, this technique gives information correlation between heteronuclei, that is proton and carbon, that are 2 or 3 bonds apart from each other. Outlines of the lecture or the course. Some silent feature of HMBC would be discussed briefly in this lecture. Detailed description of the technique would be achieved in the later on lectures of this online series. Then proton and carbon-13 NMR spectrum of some simple organic compound would be discussed, because it is necessary to know about this technique before going into the detail of HMBC. As HMBC spectrum comprises 2 axes, as it is a 2D technique. The x-axis consists of proton NMR resonances, and the y-axis of the spectrum comprises carbon-13 NMR resonances of the compound. So you won't be able to understand the HMBC spectrum without knowing about proton and carbon-13 NMR spectra. And then some spectrum, HMBC spectrum of simple organic compound would be discussed, and at the end would be an assignment to know how much understanding has been achieved about the technique. Okay, aims and objective of the lecture. After having read this lecture, the student will be able to know the basic principle of HMBC spectroscopy, and they would be able to know how this technique is helpful in structural resolution of simple organic molecules, and to determine the structure of organic molecule using HMBC and other NMR techniques. Okay. These techniques normally provides a correlation between proton and carbon-13 that are 2 or 3 bonds apart from each other. Correlation of 4 bonds and 5 bonds also appear sometime in the spectrum, but such cases are very rare. And this technique is used for detection of quaternary carbon. It links separate structural fragments obtained from analysis of HMBC, etc. The HMBC, as already discussed, this spectrum is normally plotted with a 1D proton spectrum, one-dimensional proton spectrum on x-axis, and one-dimensional carbon spectrum on y-axis, to show cross-peaks or correlation that are observed in the 2D spectrum. There are certain limitations of the technique as well. For example, sometimes some 2 or 3 bonds correlation are extremely weak or not appear at all, and this misguides us because we assume that such connectivity or interaction are not present, but the interaction is actually present. So it should be borne in mind that the absence of an HMBC cross-peak cannot be taken as an evidence against a specific structural connectivity. There is no simple method to differentiate between 2 or 3 bond correlation. This is another drawback of the technique. And in case of weak HMBC, correlation can't be decided easily whether the cross-peak is due to 3 bond or 4 bond correlation. This is the proton NMR spectrum of ethyl propanoate. In this spectrum, we see 4 peaks that are due to the presence of 4 different types of proton in the molecule. As we know, proton NMR gives us 2 basic information. Number 1, number of protons that are present in the molecule, and number 2, types of proton, that is how many different types of proton are present in the molecule. As the molecule is having 4 different types of proton, so we are having 4 peaks or 4 resonances in the proton NMR spectrum of the compounds. Okay, the peak in the downfield region of the spectrum, that is at 4 ppm, integrating for 2 proton can be assigned to the oxymethylene proton because these are the most deshielded proton in the molecule. Similarly, the peak at 2.4 ppm resonating for integrating for 2 proton can be assigned to the methylenic proton attached to the carbon having connectivity with the carbonyl carbon. Okay, the rest of the peak are peak, that is these 2 peaks, are due to these methyl protons. But we can't say precisely which methyl proton has given rise to which peak, or we can't say that which peak is due to which methyl group. So in such cases we will look for HMBC spectrum to show the connectivity and to solve the problem. This is the carbon-13 NMR spectrum of the compound. In this spectrum, we see a peak in the downfield region, that is at 174.4, that can be assigned to the carbonyl carbon being the most deshielded carbon in the molecule. Similarly, the peak at 60.2 can be assigned to the oxymethylenic carbon, as this is also deshielded carbon. The methylenic carbon that is connected with the carbonyl carbon show resonances at 27.7, and the 2 methyl carbon appeared at 14.3 and 9.1 in the spectrum. So when the HMBC spectrum for this compound was obtained and read, it was concluded that the peak in the downfield region at 1.3 ppm was due to methyl attached to the oxymethylene. As the spectrum show connectivity of this methyl group resonating at 1.1 with the carbonyl carbon and with the methylenic carbon having a peak at 27.7. Similarly, the methyl group in the proton NMR spectrum having resonance at 1.3 ppm show connectivity with the oxymethylenic carbon. This clearly show that this methyl group is attached, having resonance at 1.3, is attached with the oxymethylenic carbon. So this technique has resolved the problem, has solved the problem, and this technique is usually used for such type of problem to show connectivity of the otherwise separated fragment in the molecule. This is the HMBC spectrum of a compound that is ethyl pentanoate. In this spectrum, the compound has 6 different type of proton. As we look at the compound, this has 6 different types of proton. So the spectrum has 6 resonances on the x-axis in the proton NMR spectrum of the compound, and 7 resonances in the carbon 13 NMR spectrum of the compound, as the compound has 7 different types of carbon. So the y-axis of the spectrum comprises 7 resonances. And the spectrum has HMBC spectrum. This is HMBC spectrum of the compound. The spectrum has almost 15 cross peaks in the area. The cross peaks are connectivity. Okay. Proton 1, that is H1. If you look at the structure, proton 1 show connectivity with C2 and C3. Similarly, H7 show connectivity with C4, show connectivity with C6. Sorry, H7 show connectivity with C6. Similarly, H2 in the molecule. This is H2. These protons show H2 show connectivity with C1, C3, C4. Similarly, H3 is the spectrum show that H3 is attached with 2 bond or 3 bond separation with C1, C2, and C5. Similarly, H4 show cross peaks with C2, C3, and C5. And H6 show connectivity as cross peaks. These are called cross peaks or connectivity, because when we proceed in the downward direction, that is in the y-axis direction, and in the x-axis direction, these 2 line intersect each other. This is cross peaks. Okay, H6 show connectivity with C7 and C5. The most important cross peaks or connectivity is in this spectrum is that of H6 with C5, because this links the 2 fragment, otherwise separated fragment of the molecule. That is, it links this proton with this carbonyl carbon, and the otherwise through this oxygen bridge. Okay, this is the HMBC spectrum of ethyl benzene, a very simple organic molecule. In this spectrum, we see that these metal proton, that is H2, is connected with carbon 1. This metal proton is connected with carbon 1, that is, this show connectivity with carbon 1. These metal proton are connected with the quaternary carbon, this aromatic quaternary carbon, this HMBC show this connectivity. Similarly, H1 dash, this benzylic proton, H1 dash, this benzylic proton have this benzyl if we proceed in the downward direction, then the HMBC spectrum of the compound show that this benzylic proton is connected with C2, this aromatic carbon, and C1. This benzylic proton is connected with C1 and C2. Similarly, the aromatic proton, that is H3, H2, H4, these aromatic proton H2, H3, H4, H5 and 6, these are equivalent protons, so they have labeled only proton on one side. Okay, these aromatic proton show connectivity with the carbon in the ring, with the aromatic carbons. So in this way, HMBC spectrum show how the proton are connected with the carbon through 2 or 3 band separation. Okay, these are the references for the lecture from where the material have been obtained, and at the end, we as we discussed in the outlines of the lecture, I have an assignment for you, and the assignment is that you have to search for in the natural product glycosides. Natural product glycosides are those compounds. Natural products are those compounds that are isolated from natural sources. These are secondary metabolites, and some of the natural products have glycone portion, that is glucose, fructose, etc. attached to them. So these include, for example, flavonoid glycoside, lignin glycoside, and terpene glycoside. So you have to search for to which portion or to which carbon the glycone portion, that is etc., is attached with the A glycone portion, and whether through oxygen bridge or directly. Okay, this was all about this lecture. Hope to see you soon in the next lecture of this series. Okay, bye.